the MIRCA is a microfluidic device that is designed to assess the red cell's flexibility. And at first, it just assessed the red cell's flexibility under regular oxygen levels. But since our intended use was for sickle cell, it was important to be able to stress the red cell out more, reduce the oxygen levels so that we could get polymerization of the abnormal sickle hemoglobin and, look at the red cell under both hypoxia and normoxia. So we used chemical hypoxia because it's an open system, and to also make it, smaller, more nimble, so there wouldn't have to be this big nitrogen tank, which is usually how we induce hypoxia in the red cell. The red cell is kind of forced through this gauntlet of pillars that get closer and closer together. And when you've gotten the red cell stuck because you form a circuit, that's the impedance part. And so this is a simple device that doesn't require microscopy or anything like that, because you don't have to visualize that it's blocked. You can detect it through impedance. There is a sort of similar device that's commercially available called the LORCA. It's kind of funny that they sort of rhyme with each other. That does use gas hypoxia. It does require a nitrogen tank. It's $85,000. The reagents are several thousand more a month. And it's not measuring the red cell's flexibility mechanically like the MIRCA does. It has the red cells sort of floating in a viscous solution, and it stresses them through kind of a difference in the salt level of the solution. And the change in the red cell shape is detected by a laser. So it's a lot more involved and hard to use. I've been using the LORCA for about 8 years, and me and others in the field have become increasingly frustrated with it because you don't get the same value running the same sample on two devices sitting next to each other. And the values are changing over time as the company tweaks the device. And so as somebody focused on sickle cell disease that knows we really need to be able to reliably and longitudinally assess a person's red cell quality, these were concerning. It was difficult to run a clinical trial if your LORCA broke, they couldn't fix it, they had to give you a new one, or they changed other aspects of the reagents, and your value from earlier doesn't correspond with the next clinical trial visits value, not because of drug, but because of technical reasons. So sometimes simpler is better. And mechanically measuring the red cells quality eliminates some of the artifacts that the LORCA has. It's possible that a really poor quality red cell kind of spins on its own axis and you don't see it at all based on how the LORCA is designed. You would capture that using the MIRCA. We developed this device in collaboration with Case Western Reserve. Umit Gurkin and Pedram Mohsini are the major contributors. They're both engineers. I'm their hematology partner, but I didn't just, send them patient samples or, run patient samples and report to them. This was a true collaboration where we met every 2 weeks, discussed the device's performance, what does it need to do, where does it fit in the clinical space. And so it's been a really great collaboration, and the contribution rose to the point where I'm an inventor as well as the engineers, as is this really talented undergraduate from Georgia Tech that has been working on the device for the past 2.5 years, Akshay, who's the first author on the paper, as well as another Georgia Tech undergrad that he kind of brought in and has been mentoring, who's a year behind him at Georgia Tech, Nav. So it's been great to help, mentor both of them. We've gotten this device to the point where, we've run lots of samples on it. We've done the analytic validation of knowing, what coefficient of variability you get. It's excellent. Chip to chip agreement is great, so it doesn't have the problems that the LORCA has. We've done the clinical validation, so the values that we get from the device correlate with patient outcomes, with changes in their therapy, and if there are no changes in therapy or in their clinical status, the values are quite stable over time. So those are all the kind of things you want a device that gives you biomarkers to have. You want them to, be a precise device with little variability from run to run, operator to operator. You want it to have clinical significance. It has to, changes in those values need to mean something. And you want it to be stable over time if you haven't changed the person's medications anyway, or they have not become sick or worsened in any way. So that's a nice, smooth longitudinal assessment that we've done. But we found out something else that I hadn't expected. So when you transfuse a person with sickle cell, which is common therapy, sometimes patients are put on it every month. Sometimes they get it for an acute problem like acute chest or aplastic anemia while they're in the hospital. When you look at a transfused patient sample on the LORCA, it averages together the healthy red cells and the person's own sickle red cells. And so you lose kind of the signal of what their red cells look like. And this is a problem when you're doing a clinical trial of a drug, for example, and you've been following them and you want to see how their red cells are changing on the drug. If they need a transfusion, then you lose the data point and you can't really assess them until that transfusion has left the body, which can take up to 3 months. Another situation is if you enroll a patient on a gene therapy trial and they're already on chronic transfusion therapy, or they've been put on it in anticipation of going on the trial. You can't see what their red cells look like at the start to be able to judge how much the gene therapy improved them at the end. And I've tried lots of ways to separate out the hemoglobin A, the donor blood from the recipient blood, gradients of different densities and things like that. And it was time consuming and didn't really work. And so I was actually really surprised when we looked at the MIRCA data and we arranged samples based on the percent sickle hemoglobin present. And so where the sickle hemoglobin was very low because the person was transfused, all the way through to somebody who wasn't on any therapy, gave us this wide range. And we expected to see, like with the LORCA, kind of an averaging of the donor blood and the recipient blood. But instead, we got a U-shaped curve. And so the MIRCA was actually seeing the endogenous cells preferentially, the person's own cells preferentially, even when they were heavily transfused. And this was really exciting because it opens up a whole new avenue of not having to throw away a time point or multiple time points on a clinical trial if the person gets transfused, of being able to see those natural red cells in somebody who maybe is going on gene therapy and has been chronically transfused for years. And then another potential indication, chronic transfusion therapy, can make patients feel a lot better, but it's not perfect. It's not a cure. There's still 30% or more of the patient's own cells, which can sickle and cause pain, cause organ damage. But now that we can identify that population, we can experiment with what drug therapy can target that remaining 30%. And you would never get to see that with the LORCA because of that averaging effect and inability to isolate and actually study those natural sickle red cells. So we didn't expect that to be what we would find. And it's been really exciting and very timely discovery to make as we start to explore, dual transfusion and drug therapies for patients and become more and more involved in gene therapy and have multiple different gene therapy strategies that we need to be able to compare head to head. And so our patients are not all the same. It's not like this is what a sickle red cell looks like. This is what a healthy one looks like. It's this very personal, individual disease. And so you really need to know where the patient started from to be able to assess how much improvement they got from a given therapy. So being able to reclaim that information, even in a heavily transfused patient, is really important.